Okay, guys, so the last set of notes for this section is, and let me go back to the first slide. Here it is. Okay. We're going to talk about the technology of how scientists go and study the DNA, and then also one other component, and this is what this slide is all about, is that your genes matter, your DNA matters, but your genetic makeup is also influenced by the environment around you. And that's what this is saying. So just because it's in your genes doesn't necessarily mean that it's going to be the outcome. Or if it's not in your genes, it doesn't mean that it's not going to necessarily change as you continue to grow and age based on your environmental factors. So, for instance, here this is highlighting the turtle. When the turtle lays the eggs in the sand, they are sexless at that point. Their gender has not been determined yet. And based on the temperature of the sand, that determines whether the eggs then will become male or female. And you can imagine that within the nest that she laid, eggs that are at the top are going to be a little bit cooler in temperature, being closer to the surface and the environment. And eggs at the bottom of the nest, all that extra weight, all that extra, packing together, they're going to be a little warmer in temperature, so that affects their sex outcome too. So that's kind of a cool statistic. For those of you that have had maybe a sociology class, you might have also heard about the really sad Russian study that was done like back in the 50s. It was done in Russia, obviously not here. And it was an orphanage where there were twins, and they were comparing nature versus nurture. And so remind me tomorrow, and we'll talk about that in class, because that's kind of a sad study, but it also shows you how environment can impact your phenotypes as well. As you can imagine, if you don't have access to good nutrition and good health and good medical care, your health is going to be compromised, and that's going to have an impact on your longevity and your health as well as your DNA. All right, so then let's talk about technology. I showed you this in class today, and we talked about pedigrees. Pedigrees are a way that geneticists, it's usually the first step that geneticists take in trying to figure out a family, passing on of traits. They're going to look at the physical characteristics of you and your generation, and then your parents' generation, then back to your grandparents' generation, and as far back as they can based on that. Now, they're going to look at some that are just traits that are not tied to the sex chromosomes, and they also can look at the ones that are tied to the sex chromosomes, like hemophilia, color blindness, muscular dystrophy. Those are all tied to the sex chromosomes, and they can look at those. And notice again how the alleles are actually expressed like an exponent when you see them on the chromosomes. And in this case, capital letter is the dominant, and the lowercase letter is the recessive, as we've been expecting. Remember, a slashed means it's a carrier, so it's automatically going to be heterozygous. And you carry the gene, but you yourself do not have it expressed. And over here, what was I going to say? Sometimes you don't know whether that person is homozygous or heterozygous. Yes, you know it's dominant, but you don't know if that dominance is because of a homozygous situation or a heterozygous. So it's a kind of a mystery puzzle, and sometimes you might have multiple options. All right, karyotypes I showed you in class. You did the activity today. So the only thing that I want to really focus in on is you have your 22 pairs. Those are your autosomal chromosomes. They give you all your physical characteristics. You have your 23rd pair, which is your sex chromosomes, which determine your sex. Okay, and remember, some traits are polygenic, which means like here, this segment right here might code for eye color, and this segment might code for eye color, and this segment might code for eye color, and this. So those are polygenic traits. They're present on lots of different genes, and that's why there's so much variation in our eye color, our hair color, our skin color, and so forth. All right. Genetic screening. How do scientists now actually work with the DNA to determine things? We know that because of the Human Genome Project, they map the location of every single gene on every single chromosome in the human genetic makeup. And doesn't mean we know what to do with every single one yet, but that's what they're still experimenting with. But we know where they're located, and that's the first step in helping to solve things. So some of the things they can do is they can go through and they can determine risk factors. They can determine whether you are predispositioned for something. Like we talked about Parkinson's in class today. They can actually process your DNA and see whether you have the gene for that, or whether that gene is missing, and you might get it or you might not get it. and they can start like predicting your future. Now again, that environmental thing also comes in there. So they may say, oh, you have no problems, you're gonna genetically, it looks like you're gonna live a long healthy life, if they could say that completely, which they can't 100%. But then you're a smoker and a drinker and a partier and you live a bad nutritional life, you don't have good health care, you're not probably going to live a long life. So environmental factors play into that too. But with a simple little swab of your saliva to get some of those cheek cells, you can have it sent off and you can have it processed. And we talked a little bit about that in class. For as little as like $99, you can get some of your basic genetic information sent to you. And there's like a lab over in Greenville, South Carolina, we talked about in 4th block, that does something like that. Lots of websites if you want to decide at some point in your life you want to do that. Remember, it's kind of a catch-22. Maybe you want to know your destiny, maybe you don't want to know your destiny. Okay. Then there's you watch like your CSIs on TV and you see how they're using DNA to actually catch a criminal. Well, this is an example, and it involves DNA fingerprinting. Okay, or you watch the Jerry Springer show and they're like, oh, who's the baby's daddy or whatever. They're doing these kind of tests in order to figure that out, and it's called DNA fingerprinting. What they do, and they generate something like this, but let me backtrack as to how do they do that. So this was the slide that I started with. They put in what's called a restriction enzyme, and it's like basically they call it molecular scissors, because it's going to go in and it's actually going to cut the DNA into segments. Then they put it into a solution, and they run that solution through. This is like a jello-y. That's why it's called gel electrophoresis. It looks like jello, clear jello. They put it in there, and I'm going to show you a video clip on this on Friday. And they hook it up to electricity, and it causes the DNA to move. And longer strands only travel a short distance because they're heavier, because they're longer strands. And short strands travel a longer distance. If let's say you have a criminal then, oops, I went to the wrong slide. If you have a crime scene where you get like a suspect's DNA, you can compare the actual criminal's DNA, but you don't know who the criminal is, you can compare it to all the suspects and you can see which one matches. In this case, they're looking at a mom and a dad and a couple children to see where the genetic markers are. For the baby daddy situation, they get the daddy's DNA, they get the baby's DNA, they get the mommy's DNA, and they look at them to see, okay, the more striations that you have that line up, that means the more likely they are related to each other. So that's basically how they determine baby daddy. Okay. Next thing is talking about genetically modified organisms, and GMOs come in plants, animals, everything around you is being genetically modified. I mean, even us as humans are being genetically modified in a good way for the most part. So I'm going to show you some video clips on this. So I'm not going to talk a whole lot of notes to you, but what does it mean? It means that we've gone in and we've genetically changed the DNA to what we think is the benefit, but because we don't know what the long-term effects are, we may actually be causing more harm. And that's where the controversy comes in with genetically modified organisms and people who choose to only be organic, meaning they're not eating anything that has been genetically modified, versus those of us that are just eating general stuff off the shelf that may have been, or most likely has been, genetically modified. All right, and we're going to talk about that in greater detail tomorrow when I show you some video clips as well. All right, last slide is talking about gene therapy. Gene therapy is one of the specific ways that we are scientifically going in and changing the genetic code. So we can go in, and if genes are missing, we can insert them into a virus, put that virus into the person, those genes then get infected into the cells, and hopefully cause a positive outcome. And I'm going to show you some video clip on that tomorrow as well. But gene therapy is one of the ways that we are actually changing the genetic makeup to replace defective or missing genes, or to add in ones that are, or basically get rid of the ones that are not. So gene therapy is a very, very cutting edge kind of technology with our genetics. So anyway, those are the notes for tonight. Be prepared for a quiz tomorrow, and I will see you in class. Bye.